Hi, my name is Lenore Reiser from AgBioData, and I'm going to make a couple of quick announcements before we get on with the talk for today. So the first thing I wanted to mention is that we have a Slack channel that we would like to open up a workspace for the general AgBioData group. So we're currently working on a code of conduct that would go along with that. And if there's people who are interested in participating in that Slack channel, I guess just please, let us know. We'll make that available to you along with those sort of general principles about how we want to manage that group. Another thing about communication is that we have, as you know, this mailing list. And this mailing list, we want to remind you that if you have anything that you think is of interest, that you want to communicate with the general AgBioData community, please go ahead and you send that to the mailing list. There is a moderation, so we'll just check and make sure about that the content is appropriate, but they will be released in a timely manner. So if there's any like workshops or presentations or conferences that you'd like to share with the group, please go ahead and email that to the group. And speaking of conferences, AgBioData is going to be hosting a bioinformatics workshop. Marcella and Shabri from Phoenix are going to be organizing that. So the workshop will be to introduce attendees to a variety of digital research resources. So if there are people who are planning on attending ASPB and think that they might have something of interest in that group, please, I guess, contact Marcella and share your abstract. So I think, Marcella, you're pulling the speakers from the submitted abstracts, right? Nod your head.
That's the idea. We don't have yet a date, nor an allotted time, so we don't know. Last year, we accommodate everyone that had an interest in participating, but that led to very short talks. They were like 7-8 minutes or something. So it was like speed databasing. So we don't know yet how much time we're going to have this year. Once we have that, we'll decide the format of the meeting. We just know that it's going to be fully virtual, and we'll try to accommodate as many resources as we can.
Okay, so get your abstracts in. Okay. And then before I forget, our next meeting will be on May 5th, again at 10 o'clock. And Monica Polchow is going to provide us with updates from the GFF3 working group. So with that, I'm now going to move on, and it's my great pleasure to introduce my colleague and friend, Peifen Zhang. So Peifen did her PhD at Iowa State and postdoced over in Albany with Barbara Baker. She and I met when she was the director of the Plant Site Project at the Carnegie Institution, and she's now a project scientist over at Phoenix Bioinformatics. And today she's going to present the project that she's been working on, which is PhyloGenes, an online resource for a free online resource for understanding and predicting functions for plant genes. I'm going to ask if you have questions to put them in the chat, and then we'll, if there's, we'll have some time at the end for her to address those questions. So if you just give us a little bit of a minute, Darwin is going to be running the slides, and then I will be running the demo. So I'm going to turn myself off now.
I wanted to thank the meeting organizers. Thank you for the great opportunity to let me tell everyone about PhyloGenes, and thank you everyone for showing up. So in my presentation today, I will start with a few PowerPoint slides to give people a very brief background on phylogenetic function inference and the basics about PhyloGenes. After that, I will switch to a live demo to just to show you how to find the data and navigate through the PhyloGenes website. So From a sequence to function, the fastest way is to run a computational prediction. The most popular among all the methods, the most popular ones are sequence similarity based, including BLAST and interpro-scan. Another method that relies on the evolutionary relationship of homologous genes in function prediction, that is called phylogenetic function inference. In this toy example, I'm showing you that a group of homologous genes, their evolutionary history has been depicted in the form of a gene tree. On the tree, the speciation nodes are colored in green, the duplication nodes in orange. Any pair of genes are related to each other either through a speciation event or through a duplication event. Orthologs are a pair of genes that are related by speciation. So as you can see in this example, paralogs are related by duplication. Paralogs can be within the same species as in this example. So we see mouse gene 3 and mouse gene 3 prime. They are related by duplication. Paralogs can be found between species. So in this example, the mouse gene 1 and human gene 2, they are paralogs because they are related to each other. You can trace back to that orange dot, a duplication event. One of the principles that supports phylogenetic function inference is the belief that protein function changes rapidly after duplication. That leads to paralogs share different functions. The orthologs return the ancestral function. In another word, orthologs would be most likely to share the same function than the paralogs. So going back to this toy example, if we can, in this example, we can see that we know the function for the mouse gene 1, and we know another function for the mouse gene 3. Applying the ortholog conjecture, we could predict the function for human gene 1. We can also predict the function for the human gene 3 based on ortholog conjecture. As for human gene 2, we don't know the function of its ortholog, which is the mouse gene 2. We only know its paralogs in this gene family. So when we predict a function for human gene 2, we need to be extra careful. We probably need to look into other information such as the sequence alignment and the gene expression, etc. So this is a quick recap. Phylogenetic function inference is to assign function from a characterized known gene to an unknown gene using the evolutionary history of the genes. So there are two key points here. First, we need to find so in order, to find a function for our gene, we need to first find its orthologs. And we need to know function for at least one of the orthologs. So PhyloGenes is a free web resource that you can see the URL here, and this is the homepage. PhyloGenes displays pre-computed gene families and gene trees, and currently it includes 40 plants and 10 non-plant model organisms. The gene families and the gene trees are derived from Panther database, and one protein sequence per gene was used in building the families and the trees. Right next to a gene tree, PhyloGenes also displays experimentally validated gene function information of the family members. The goal was to help scientists predict functions for the uncharacterized genes. The project is developed by Phoenix Bioinformatics with collaboration with the Panther Group at USC, and it was funded by NSF for 2 years. Okay, so this is a typical gene family page. It has 3 parts. The family info band has very basic stacks, such as the number of genes in the family, list of organisms represented here, and the taxonomic range of the organisms. The left side shows the gene tree. By default, it's in a compact view. the purpose is to highlight the genes with characterized functions indicated by a yellow flask icon on the tree. And of course, you can expand the tree. On the right side is the gene table, which provides information of each gene in the family. Each row is a gene row. It's aligned with the corresponding gene in the tree. And you can see here molecular function on the header among the columns. F stands for molecular function, P stands for biological process. Those are Go annotations that are supported by experimental evidence for at least one member of the family. Each subheader under function and process is a unique Go term. And if you see a yellow flask in the gene row under a particular Go term, that indicates the presence of that function in that gene based on experimental evidence. And in this real example, I'm showing you a gene family page on PhyloGenes. And I wanted to, using this real example to show the value of PhyloGenes. So let's assume that we wanted to find functions for this grape gene. We found its Arabidopsis ortholog, which is characterized as long-chain fatty acid-CoA ligase. You can see the header in the gene table column. Applying the ortholog conjecture, we can predict that the grape gene is most likely to be a fatty acid-CoA ligase. There are other genes in the same family that share the same function, so they are fatty acid-CoA ligase by experimental evidence. We can, based on those characterized genes, to trace back to the most recent common ancestor of them. So that's the most recent common ancestor of this function, fatty acid-CoA ligase. We noticed that this family has multiple functions. So from the header, you can see there's 4 coumarate CoA ligase. So there are genes in the same family that carry on that function. Similarly, we can trace back to the most recent common ancestor of 4 coumarate CoA ligase. The gene tree shed light on the evolutionary history of these 2 functions, how they relate. So you can see that. You can tell from this tree structure that the fatty acid-CoA ligase function is more ancient, and the 4 coumarate CoA ligase function evolved from it after this recent duplication event. There is a red mark pointing to that. So we learned the basics about PhyloGenes. Now I wanted to switch to our production website to use web demo to show you how to find the data and navigate through the website. This is the homepage. There are 2 search boxes, one on the top, one in the main page. You can use either one. And all search will return gene families. So keep in mind, we don't have gene pages, we have gene family pages. And it indicates in the search box that you can search with different ways. You can use UniProt ID, you can use gene ID, gene symbol, or a keyword. Currently, it only works with exact match. So, which means that if you have a typo or you put a partial word, it's unlikely to return anything to you. But let's try with this gene ID. So Lenore put in this gene ID, click on search. Right, so we come to this gene family page directly because that's the only matching family to that gene ID. On the left side is the tree, right? You can navigate the tree by panning it left and right. You can move it left and right, and you can scroll up and down. And when the tree moves up and down, the gene table also moves along. So the gene row in the table is always aligned with the corresponding gene in the tree. Yeah, great. And if you can show the family info band. Right now, I don't see it. If you can scroll a little bit up, that we can see the gene info band. There you go. So you see that the number of organisms is underlined. It's a hyperlink actually. Yeah. And that lets you to prune the, if you just click on that. Yes. So it lets you to prune the tree. I strongly encourage people to use the tree pruning tool. It lets you to focus on the genes and the species of your interest. And it also speeds up the page load. So I think Lenore just did a single click to deselect everything on the very top, right? Yes. And then you can select, yeah, you can select all, you can deselect all. And then let's say we deselect everything. We select Arabidopsis, maybe Brassica naba, maybe a couple others. And then you can update the tree. This tree pruning is working on the fly, so it will take a little while. Right. So now we are looking at a pruned view of the same gene family tree. Only genes from the selected species are displayed. We can expand to see them all. So there you go. In a single click, we can see all the genes. Okay. So now look at the right side. That is the gene table. Yes, and so you could scroll horizontally to see all the columns in the gene table. There you go. And notice that it indicates there are hidden columns right next to the configuration icon in the header of the gene table. It says there are 3 columns hidden. So yeah, there you go. If you click on the 3 columns hidden, it brings you to this customization tool. Customization lets you hide, show different columns and reorder them if you like. So customization is very helpful. It will let you focus on the information that is most important to you. And sometimes it's necessary to reduce the table. If the table is too large, the loading will be very slow. So that's the customization part. And great. So Lenore is showing you the customization. Okay, great. So there is this operation menu right above the tree. Yeah, we let users to download the data from PhyloGenes. You can download the whole gene table in text format. You can download the tree in several formats, including image and including PhyloXML. And you can download a list of plant orthologs of your gene. To do that, you click on the gene. Yeah, so that's an instruction. You click on your gene from the tree, and then you click download plant orthologs. The ortholog list is generated on the fly. It may take more than a few seconds, so please be patient. Once the file is ready, so can we see that download pop-up window if you still have it somewhere? If not, maybe just click again and then click that wait, don't do anything yet. So it's trying to generate the list for you. There you go. Once the file is ready, then you click that, the file is saved to your computer. Lenore, can you do one more thing? I wanted to show people. So the search box you see on the top, right top corner, the search box, if you don't put anything in the search box, but click on the search, that will generate a list of all the gene families. So that's I think right now over 8,000 gene families. On the right side, the navigation area, there are 2 filters to let people narrow down to the list of the search results. One is taxonomic range. The other is organism included in the family. So for example, we are interested in finding all plant-specific families, you could choose, I think Lenore is choosing Umbreophyta, that's fun. So that will show you list of families that are specific to Umbreophyta. If you are interested in families that has at least one gene from, let's say, Arabidopsis, you could choose, you could click Arabidopsis in the organisms filter. So that's the filters. Yeah, great. Thank you so much. So sometimes you can't find your genes. Keep in mind that not all genes from an organism are part of gene families. It could also be that your species of study is not included in PhyloGenes. In that case, go back to the homepage, make sure that you check out this species list. If you don't see your species there, click on that button. And Tree Grafter is a tool that lets you to copy paste your protein sequence, one sequence at a time. It will try to find the best matching family and place your gene in the proper location on the tree. It will be labeled as grafted. I gave you a brief background of PhyloGenes, and I gave you a brief demo on how to navigate the website and find the information. So hopefully you'll find it, PhyloGenes will be useful to you or to your users if you are database developers. So for database developers, here I'm showing you a way to link out to PhyloGenes gene trees. So that's that URL given there. And we will be more than happy to work with you to make sure that the gene IDs are the right format in order to link out to PhyloGenes. So I'm showing you one example implementation right now we had at TARE. So we have two ways to link out to PhyloGenes. One showing their added, so that's a link we add to all genes. The one at the bottom is a type of link that we only add to genes if the gene is part of a gene family in PhyloGenes. If the gene is not part of PhyloGenes, we then indicate that on this gene page, focus page. So there are two ways to link out. I'm showing you here. I think that's my last slide. So this is a beautiful family behind the PhyloGenes project. And thank you everyone for listening, and thank you everyone. And if you have questions, please feel free to ask. We still have plenty of time.
Peifen, there's one question, which is, are there any plans to continue updating data after the project funding has ended?
Right now, we're trying to renew our funding from NSF, and in the meantime, we have minimum funding from our own organization. So Phoenix Bioinformatics is supporting the project. So we are still developing it, even though right now we don't have funding from NSF. So we develop in a slower pace. We are hoping to get the renewal from NSF, and yes, we hope to continue to develop this resource. There are a lot more features we wanted to add. Those features are requests from our users, and yes, we hope we can continue to develop this resource.
So another question from Doreen about how have we compared the trees? Have you compared the trees to other phylogenetic trees at places such as Grameen or Ensemble genomes?
Systematically, I can't say that. So maybe let me go back to the how the PhyloGenes trees were constructed. So the trees, as I think I briefly mentioned, they are derived from the Panzer project, right? Panzer database. They using their own in-house pipeline to build the trees. So first of all, the families were built from HMMs, libraries, and then the tree structure were constructed based on two rules. The speciation nodes, they use NCBI species tree to guide this, to place the speciation nodes. And then for the gene duplication nodes, they use maximum parsimony to help them to place the duplication nodes. So in general, the total, the overall topology of the tree, of the gene tree, mostly they follow the species tree, and the duplications, those are placed at maximum parsimony. So in a way, the topology of the tree is very robust, and compared to other tree topologies generated using different pipelines, I'm not sure there. I can't speak for that. I didn't do a systematic comparison based on my personal experience. Looking at a few genes, I'm interested looking at the tree topologies at ensemble plants and at plaza. Often they are more or less similar, but systematically I can't speak for that. I think the paper published by the Panzer group, they developed the algorithm called Giga that they used to build the trees. In that paper, they did some large-scale analysis. So that's all I can say.
So another question is there a way to visualize multiple sequence alignment? So if you want to look and compare. Okay.
Yes, I forgot to include that part in my demo. If you go to a gene family detail page, on the top of the gene table, there is a little link. You can toggle between text message of the genes versus the multiple sequence alignment. Yes, you can easily toggle to see multiple sequence alignment in that gene table, which is right next to adjacent to the tree, and it's aligned with the tree, so you can easily compare.
Thank you. And there's two related comments. So one is very nice, love the tree grafting feature. And is the tree grafting based upon BLAST-P? How does the tree grafting work?
Okay, good question. So tree grafting has two steps. The first step is that it takes your sequence and then try using the HMM and try to place your sequence to the right HMM library, and then it will rerun multiple sequence alignment to put your sequence to that MSA for that gene family. And then it runs, use this algorithm called REX-ML. So it's maximum likelihood. It's a very popular algorithm, REX-ML. And then try to place your gene to the existing gene family tree, but with a constraint to try to not to change the overall tree topology. So the grafting result, you can see your sequence inserted to the gene family without changing the overall tree topology. So did I answer your question? So if you go to the PhyloGenes site trying to do a tree grafting on that sequence copy paste page, there is a link to the published paper on the tree grafting algorithm. So keep in mind that the goal for the tree grafting tool is for function inference. So it tries to place your gene into a location, the best possible location, so that you can infer function for your sequence. But in several cases that I noticed is that it does not completely put the right evolutionary history for your sequence. I'll give you one example. I'm sorry that you can imagine that in your head is that your sequence is from, let's say, Snapdragon. And after this tree grafting run, your sequence is put into a recent duplication node of Arabidopsis. So you may see that your Snapdragon sequence is placed under a duplication node of, let's say, 4 Arabidopsis genes. So evolutionary speaking, it's unlikely because those sequences from different species, but it tries to put your Snapdragon sequence into the location so that the parent term, the parent speciation node will help you to do a function inference.
So thank you.
So, yes, yeah. I'm
Sorry.
not sure if I.
Are you ready for
I
another question?
Yeah,
I think
I'm not sure if I. Okay. Okay, go ahead.
Yeah, I think the explanation was good. So another question was, again, thank you for your nice talk. And can you list gene families by size? Is there a way to do that?
Okay. From the website, not, but if you are interested, we can easily generate a full list of the families sorted by the size. I guess the size means the number of genes in the family. We could, we could help you, with customized information or customized list of gene families, but on the website, there's no way right now.
And another question is, if we want to hyperlink to either the tree grafter or the gene family, is there code that checks to see whether or not a gene is within a family or not? So what happens if you pass a identifier and it's not present in the family?
Yeah, very good question. So right now, if you put a identifier that is not recognized, you will see like an arrow message on the page, right? Your gene cannot be found. Is there a way to for the developers to check? What we can do is that we can generate a complete list of genes for your species that are part of PhyloGenes families. Maybe you can use that list to look up. Or if the interest is high, we could develop an API for people to to call the API and then see if a particular gene identifier is part of PhyloGenes families. But either way, please feel free to write us an email, either write email to me or to Lenore or just to anyone at Phoenix Bioinformatics. We'll be more than happy to work with you.
So info@phylogenes.org.
Yes.
Put that in there, the chat. Are there any other questions that people have?
The contact information, if you go to PhyloGenes, any page at PhyloGenes, there is this help link, I think, on the top. Click on the help or click on the contact. It will, you will see how to reach us.
So one more question from Andrew. Are there trees and functional annotations available to download in bulk? Would that be the Zenodo site, to download the whole data set?
Yes. So if you are interested to download gene tables, right? So all the functional informations of genes of families are displayed in the gene table. You can download one table at a time, but I think if you're interested in download all the gene tables for all the families, we have generated a zip file, and we placed it in Zenodo. I'm not sure. We have a link from
Tanya
our
put it
website.
She put it in the chat.
Okay, and I think that's actually good idea. I think we should put the Zenodo link on from the PhyloGenes help page. That's a good idea. I don't think I have done that.
Are there any other questions? If not, then thank you. Oh, wait, here we go. Is there a way to download all plant gene families or genes of a particular family?
Okay. First, the second part, to download the genes, all genes of a particular family. So you go to that family page, right? When you download the gene table, in that downloaded gene table, it has all the genes in that family. And I believe it includes the UniProt ID of each family member, the gene ID of each family member. So hopefully that helps you. And the first part is to download. I'm sorry, I lost the first part of the question.
To download all plant gene families. So I'm assuming this means maybe just like green cut. So gene families that contain only representatives from plants.
Right. So remember using that the filter, right? If you do a blank search, you get list of all the families, and if you use the taxon filter, you can get a list of all families that are specific to Viridiplantid, for example. But then I guess you wanted to download that list, right? For example, family IDs. No, right now I think that's from the search result page. There are several pages for hundreds. If the number of the families are hundreds, those are shown on several search result pages. There's no single download utility to let you download the complete list of of families following that category. But with some of these very nice user features we have not developed yet, but we are more than happy to generate those customized data sets for you. Please feel free to contact us. We will be more than happy to help you.
I'm gonna, there's one last question, and then I think that will end the questions. And that was just one about plans, if there are any plans to incorporate non-coding genes.
Not in the immediate future. Not now. So the whole pipeline for detecting gene families and constructed trees are developed for protein coding sequences. So it's protein sequences that are used in constructing the HMM libraries and the trees. So and yeah, I don't see non-coding sequences in the near future, but maybe I don't know. Maybe I should look up other resources so that users can know where to go to find the non-coding sequences in gene families.
If there's no more questions, so are there any other announcements or bits of information that people want to share before we take off? And if not, I just want to remind people to please return next month on the 5th for a presentation about updates to GFF3 formats. And I think that's it. If you have any other questions for Peifen, I know she's very happy to respond to people by email. All right.
Thank you.
That's it. Thanks for everybody for coming today.